introduction, this author started to discuss about the title of that paper, how to achieve high regeneration of shoot. Okay, so he started with what are the factors that actually affecting explant regeneration capacity. Okay, so the first one is of course the plant material. Okay, so for plant material, he divided into 7 factors. Okay, so which are the genotype, the physiological stage of the donor, the explant source, the age, the size of the explant, okay, the position of the explant in the donor plant, and also the explant density. Okay, so these are the 7 factors related to plant material. Okay, so after that, he explained in detail how genotype can actually affect regeneration capacity. Okay, so you can see, like for example, he mentioned that dicot regenerate more easily than monocots, generally. Okay, so it is easier to do tissue culture of dicot compared to monocots. All right. Okay, and then comparing between herbaceous plants and also woody plants. Okay, woody, for example, and in general, herbaceous plants can be regenerated easily compared to woody plants. Okay, that's why macam for example, if people say that nak buat tissue culture of apa ni pokok pokok balak, okay, memang ni lah, but it be more difficult compared to nak buat tissue culture of chili, for example. Okay, so this is just to show you, okay, alright, the difference. Okay, right, so these are the example how genotype can actually affect regeneration capacity. Okay, alright. So they give example lah. For example, sugar beet, which is more recalcitrant compared to tobacco. Okay, so tobacco is easily to do tissue culture of tobacco is easier compared to sugar beet. Okay, right. So just comparison. Okay, and then he mentioned about the second factor related to plant material, okay, which is the physiological stage of the donor plant. Okay, so generally we know that vegetative segments, okay, regenerate more easily compared to generative one. Okay, so when you choose the explant, of course you have to choose from the healthy plants. Okay, janganlah tengok plant tu dia punya explant dah ada penyakit kan, so we have to choose from healthy plant. Okay, with high cell division. Okay, and we know that mature tissue, the regeneration capacity is low. Okay, so that's why we have to choose young tissue. Okay, so for example, macam kita buat tissue culture Jatropha hari tu, okay, I choose young tissue, young leaf. Okay, bukan leaf yang dah macam the color of the leaf dark green, bukan kita macam light green, macam young tissues. Okay, because young tissues have a high regeneration capacity than the older one. Okay, and it also mentioned that regeneration capacity of genotypes increases during the flowering period, for example in Lunaria. Okay, so he gave some examples. Okay, and the third one is the explant source. Okay, so he mentioned that the plant grown under greenhouse condition will give rise to better result, okay, than the one grown in the field. Of course, because dalam greenhouse condition compared to the field, because when you plant the mother plant or the donor plant, okay, in the field, the plants are exposed to organism, okay, exposed to pests. But in greenhouse condition, it's a contained environment. Okay, right? So, so these are the comparison lah. Okay, but in terms of greenhouse and field condition, the best results of explant source are obtained from explants excised from in vitro grown seedling. So for this is for example, remember that I showed you how to do tissue culture of chili, kan? So basically tissue culture chili tu kita dah germinate on MS medium. Okay, the seed we already surface sterilized, germinate on MS medium, then kita dapat seedling. Okay, so from the seedling, kita ambil the explant. Okay, so this is what we call for in vitro grown seedling. Okay, right, so this is the explant source. Okay, you boleh baca yang lain lah ya. Okay, and explant age, okay, right, like I mentioned just now, okay, you should choose young explant because rejection capacity of older plants is often low lah. Okay, so this is related to explant age. Then he mentioned about the explant size. So the explant size, not too small, okay, not too big, is the right size. For example, remember that I cut the leaf of Jacropha, okay, in square shape, okay, because larger explant having a big amount of nutrition reserve, okay, so it can easily regenerate in vitro, okay, and then the number 6 factor is the explant position. Okay, so where you're going to take the explant, whether the higher part of the explant, okay, or the lower part. You know that the higher part of the donor plant are older compared to the lower part. Okay, so so he gave a comparison with some example. Okay, so you can read. Okay, and then the last one is the explant density. Okay, so this is related to competition among the plants lah. Okay, among the explant. Okay, all right. So then another besides the plant material, okay, the surface sterilization process actually affects regeneration capacity. Okay, so we know that before we proceed with the tissue culture, we have to optimize the surface sterilization process. Okay, because this is a very critical stage. Because if this stage is not properly being done, okay, bila you culture the explant, okay, if you have the microorganism, this microorganism will compete with the nutrients in the medium with the explant. Okay, right. So so the surface sterilization is actually important. Okay, all right. Okay, so in surface sterilization process, the author actually elaborated more, okay, giving example on the effect of sodium hypochlorite, okay, at different concentration and application period. Okay, so we know the sodium hypochlorite is actually what we call as the commercial name is Clorox lah. Selalu kita pakai Clorox, okay. Alright, so the different concentration, you have to test the different concentration of sodium hypochlorite because we want to kill the microbes, but we don't want to kill the explant. Kalau tinggi sangat concentration of the sodium hypochlorite, okay, it will kill the explant also. Kita tak nak kita just want to, it should be the right concentration and also the application period. Okay, how long it should be incubated in the sodium hypochlorite. Okay, right, so you can read lah. Okay, alright. So this dia tunjuk gambar aja lah. For example, comparison between different concentration of sodium hypochlorite but exposed for 15 minutes. Okay, so you can see that the best concentration is actually 3.75%. Okay, but not 2.5. Okay, but and not higher. Okay, so this is the right concentration of sodium hypochlorite to be used. Okay, for 15-minute exposure. Okay, and also he gave an example on the effect of sodium hypochlorite solution at different temperatures. Okay, so you can see that. All right, so in the flax from seeds sterilized with 2% sodium hypochlorite at different temperature. Okay, so he tested. He gave an example. Again, of course, you know the main criteria. Okay, is the main factor. Okay, to be optimized is the culture medium. Okay, all right. Okay, so because culture medium is important because you have to optimize, you have to choose what type of medium that you want to use. It can be either MS medium, B5, N6, niche and niche. Okay. Okay, all right, and then in terms of the gelling agent, okay, whether to use agar or Vita-Gel, okay, so these are the example lah, okay, and then like for example to optimize the PGR, okay, and then the culture condition lah. Of course, culture conditions affect the regeneration capacity lah. Okay, so the right temperature, okay, the right duration, the right light intensity, the duration of the light, okay, all right. So this will affect lah, okay. And then he mentioned how to increase expand regeneration capacity. Okay, so we know that there are factors affecting regeneration capacity. So he mentioned about treatments that can actually increase, which is you can increase the tissue water content. Okay, so you can read he gave example. Okay, and it also can be done, okay, by regulating the osmotic pressure of explant. Okay, right. Okay, so this is the paper. Okay, and in the conclusion, yeah. Okay, right. So any question? I think this paper is very simple. Okay, and I think you can understand the paper. Okay, ada yang dah baca ke paper ni? Okay. No. Tak apa. I know you're okay. You're busy siapkan assignment semua Okay, tak apa. It's okay. Dr. Fahmi dah bagi result midterm ke?
Belum.
Oh, mana saya dah dapat dah. Oh, dia tak share lagi tak apa. Okay, because we have to release the CAM on the 30th December. Okay, so insyaAllah you dapat lah. Okay. Alright, but you can PM me lah kalau you nak tahu Makayu. Okay. Because he already gave to me the marks. Alright. Okay, any question on that paper? Very clear kan? Tak ada
Yeah.
Alright, clear. Okay, so I'm exposing you to read paper lah at the level of you second year ni. Dah kena baca paper lah kot Alright, so we go to the second paper on callus culture. Okay, so I go very quick lah Okay, so basically this paper talk about the applications of plant callus culture. Okay, so you know what is a callus, right? Okay, so basically, okay, when you want to get the general idea about a paper, okay, you can read the abstract. Okay, so you can see the abstract mentioned about traditional Chinese medicine, okay, and talk about medicinal plants. So you from the abstract, you know what the paper is all about. So basically, these authors, berapa author ni? Let me check. Oh, seorang. This author, okay, right? Actually, apa ni? Talking about the application of plant callus culture related to production of metabolites. Okay, so to produce beneficial compounds lah, secondary metabolites. Okay, so like I said, the introduction. Okay, and then he mentioned about how callus is being formed. Okay, which we already discussed. Okay, and then the culture condition. Okay, we also discussed. Okay, so you can read this paper to understand better. Okay, the culture condition. Okay, and then like I said, he started to mention about the production of secondary metabolites. So how in this photo, this diagram showed that callus is being induced, okay, and being used, okay, to produce suspension culture, okay, which in G you can see that callus is being transferred into liquid medium, okay, and to establish suspension culture, okay. So basically, this is the application. He concentrate on the use of callus for production of secondary metabolites. Okay, so I think, and he gave some example how callus culture are being used to produce phytochemicals. Okay, some of the phytochemicals lah, and with the name of the plant. Okay, alright. I think Jendara dah explain lah. Okay, alright. Okay, I think it's simple lah, boleh baca. Okay, alright. Okay. Okay, and then we have papers on suspension culture. Okay, which I have actually banyak lah. Okay, but actually lebih kurang sama je. Okay, we have 7 papers. Okay, alright. Okay, I'll choose one lah. Alright. Okay, we choose this one. Alamak, sorry. Okay. Alright, so this, okay. Alright, so this paper is not new because it's in 2008. Okay, I think, okay, you boleh tengok ni lah. Lepas tu the author, okay, seorang juga. Okay, so this is, if I'm not mistaken, this is from a book kot. Right, not a paper lah. Alright, so this author, okay, talk about, for example, the introduction lah, okay, about how plant cell culture are being used to produce secondary metabolites, okay, and this author divided into, for example, food additive, okay, secondary metabolite being used for flavors, fragrances, and colorants, okay, and also nutraceutical and pharmaceutical, okay, all right. Okay, so then the technique, okay, of plant cell culture technique, okay, all right, okay, and application of plant cell cultures, okay, which we already discussed banyak kali lah. Okay, so this author divides the application of plant cell culture into 3 main aspects. Okay, one is for breeding and genetics, okay, for model system, okay, and for production of secondary metabolites lah. So this is a model system ni for research lah. Okay, so they mention lah the chapter, this chapter from a book actually discuss about metabolite production by plant culture. Okay, so you know that this author concentrates on only on application of cell culture for production of secondary metabolites. Okay, so these are some example. Okay, alright. Compared to parent plant, macam they compare between cell culture and parent plant. Okay, you can see the culture plant, the cell culture, the production of metabolite higher compared to the parent plant lah. Okay, for example. Okay, and these are example of food additive from plant cell culture. Okay, so for example, anthocyanin, okay, vanillin, untuk flavors lah, colors from anthocyanin, okay, flavors, for example, from vanillin, garlic, onion, coffee, kan, okay, right. Then we have sweeteners, for example, stevoside, okay, and then the author elaborated more lah on in terms of aromas, pigments, okay. And then the next section, the author mentioned about pharmaceuticals. Okay, example of pharmaceuticals that can be obtained from plant. Okay, ada banyak lah. Okay. Okay, so then the author described about the advantages and disadvantages of plant cell cultures. Okay, so I already mentioned some of the advantages, okay, in the slides. Okay, so then I'm not so sure he or she, maybe she lah, compared the characteristics of microbial and plant cell relevant to fermentation. Okay, all right. And then how to achieve, I think this saya dah elaborate the strategy to obtain secondary metabolite from plant cell culture. Okay, so saya dah discuss dengan detail, okay, in the slides. Okay. Okay, and then how you want to select, okay, of the plant, okay, which are rich in beneficial metabolites. These are some of the strategy lah. Okay, okay, banyak sikit, tapi it's actually talking about the same thing lah dengan 6 paper yang lain. Okay, alright. Okay, so because I give you papers so that you can understand better. Okay, so any question? Okay, kalau tak ada, any question to my assignments? Are they clear?
Yes, clear.
Okay, kalau you have question lah, you can always ask me. Okay, alright. I hope you don't have problem with the the first assignment yang you kena choose plant yang you want to do tissue culture. Okay, it's a very short assignment. I think you should be able to get full mark lah. Okay, selalunya student dapat full mark lah, 5%. Even though 5 marks kan, it's actually contributing lah. Okay, and then assignment yang kedua tu on soalan on a paper. Okay, you have to jawapan dalam paper tu je, jangan jawapan daripada tempat lain. Okay, so jawapan dari paper tu saja. Okay, so the answers, everything is in the paper. Okay, and then you have 2 lab reports. Okay, I hope it's not burdening you lah. Okay. Alright, any question? Any question?
No.
Okay, boleh? Kalau you segan, you ada burning question that you malu nak tanya waktu kelas ni, you can always PM me lah. Okay, I'll try my best to answer lah secepat mungkin. Okay, alright. Okay, and then kalau you want consultation pun we can arrange. Okay, tapi please not during the exam week lah. Okay, saya rasa too late lah. Okay, kalau ni you nak, you can just PM me.